Microfluidic bioreactor for 3D tissue culture with optical accessibility. Wear suitable garments for general protection. The assembly procedure can be carried out with unsterile gloves. Position all the materials inside the biological safety cabinet. Biological safety cabinet. Pipette controller with sterile pipettes. Micropipetter with sterile tips. Sterile forceps. Sterile petri dish. Cell culture medium. Sterile bypass tube. Inlet line. Sterile syringes. Oxygenator tubes. Tube clamps. Outlet line. Sterile glass reservoirs. Sterile filters. Microfluidic bioreactor chamber. PDMS covered bioreactor gasket. Magnetic bioreactor holder. Multi-well plate with cell-seeded scaffolds. Incubator. Syringe pump. Standard optical microscope. Connect the oxygenator tubes to the syringes. Insert the tube clamps to prevent medium reflux. Lock the tube clamps and fill the syringe with culture medium. Avoid air bubble formation. Open the clamps and remove excess air bubbles. All the syringes must contain the same volume of culture medium. Take out the bioreactor from the sterile envelope. Fill the microfluidic channels and the culture chambers with culture medium. Place the bioreactor in the petri dish. Insert the 3D scaffolds in the chambers using the sterile forceps. Insert deeply the scaffolds in the chambers while avoiding medium leakage. Make sure to remember the exact chamber location of each scaffold in the bioreactor. Seal the chambers with the PDMS covered bioreactor gasket. Be sure that the gasket side covered with PDMS is in contact with the scaffolds. Assemble the magnetic bioreactor holder to seal the bioreactor. Align the holes of the holder with the chambers to allow optical accessibility. To prevent leakage during operation, the two holders must never get in contact with each other. Connect each chamber input to the corresponding oxygenator tube connected to the syringe. Make sure to connect the correct syringe to the corresponding chamber. Connect each output to the corresponding oxygenator tube connected to the reservoir. Connect the air filter to each reservoir. Annotate the content on each reservoir to ease the identification of the linked syringe. If you need to connect two culture chambers with a bypass tube, fill the bypass tube with culture medium before connecting it to the reservoir to avoid the insertion of air bubbles in the chambers. Connect the bypass tube to the bioreactor. Place the syringes on the pump and select the infusion parameters. Open the tube clamps and switch on the syringe pump. Place the bioreactor in the incubator and select the culture parameters. While closing the incubator door, make sure it doesn't squeeze the oxygenator tubes.